Welcome to the class of Microarray Data Analysis. Dear students, today we are going to learn about how we represent the data, gene expression data. In the last class, you have seen after normalization, processing of the data, expression data is shown in the expression matrix. The gene expression profile can be represented by the row vector and sample expression profile can be represented by the column vector. So now today we will learn how we should represent the data in the gene expression matrix. So expression data can be represented in the 5 ways, like the absolute measurement. In the case of an absolute measurement, each cell in the matrix will represent the expression level of the gene in abstract unit. We will see it. So it is not meaningful to compare expression level of gene across two different conditions in the absolute unit. So because the starting amount of the mRNA can be different because that is the same gene in the different conditions. So in the table 1a we can see sample expression matrix with each cell containing the expression level in the abstract unit. So here here you can see the table 1a contain absolute measurement C1, C2, C3, C4. These are the 4 experimental conditions, and in the rows we can see gene A, B, C and D. So these are the different genes. Then it is converted into the relative measurement with reference to the C4 condition. So this is the ratio C1 by C4, C2 by C4 and C3 by C4. So this is the relative measurement because C4 we have chosen as a housekeeping gene. Condition 4 is used as a reference and all other conditions are normalized with respect to C4 to obtain expression ratio. So this C4 we can say this is chosen as a housekeeping gene and reference set. So in the 1C table you can see the transform data. Means after the logarithmic transformation this data is obtained, and table 1D is shown the logarithmic transformation. This is obtained in the discrete values like 0, 1 and -1 forms. So in this table like C, in this table all the expression ratio were converted into the log2 expression value. This representation has an advantage of treating up regulation and down regulation on comparable scale, because for the up regulation we can say if that is the greater than 0 that shows the up regulation, if it is less than 0 it shows the down regulation. So in this table, in the table 1D you can see it is shown in the values of discrete. So values that are greater than 1 that is changed to 1, and genes with the values less than -1 that are changed to -1. Any value between -1 and 1 was changed to 0. So these are the discrete values which we have taken for the easy calculations. So once we have obtained the gene expression matrix, additional level of annotation can be added either to the gene or to the samples, and the function of the gene can be provided, like we can say the up regulation, down regulation, how genes are expressed, genes are highly expressed or genes are weakly expressed, and in which state it is more expressed, like in the disease status or in the normal status. So for the further analysis we can choose the method of supervised learning and unsupervised learning. So this we have discussed in the other lecture. You can watch that. So now we are going to discuss here the relative measurement of the expression. Now we will have the more explanation of this. In the case of the relative measurement or representation involving expression ratio, the expression level of a gene in abstract unit is normalized with respect to its expression in the reference condition. Like you have seen, each condition is divided by the C4, like C1 divided by C4, C2 divided by. So here, it is like that. The ratio of 4000 by 100 will lead to the same result as 40 divided by 10. So this is same as. So here you can see, this is the normalized C1 by C4 relative measurement. In the 1b table you can see, this is the example of that what we are discussing now. Thus any information on absolute measurement will be lost in such representation, but now meaningful comparison across different conditions can be made as long as the same reference condition is used to get the expression ratio, because we are using C4 in all the cases. If we take this example, the C4 is taken to normalize all the C1, C2, C3. So this will impact similarly, but this will make our calculation easy. Thus, okay, because we are normalizing all the conditions by the C4. So because reference condition is the same, okay, for all other calculations. So it is shown in the B, table 1b, you can see here. This is the relative measurement after the normalization. Now come to, this is the logarithmic transformation. In the case of tables representing the log to expression ratio values, information on up regulation, down regulation is captured and is mapped in a symmetric manner. Okay. For example, 4-fold up regulation maps to log 2. Okay, if it is 4-fold. So 2 and 4 4-fold, and a 4-fold down regulation maps to log 2 1 by 4. It is -2. That means it shows down regulation. So genes are down regulated. Genes shows low expression. Okay, that means low intensity means low expression. Thus, from this table, the fold change for a differentially regulated gene under any condition can be easily recognized. So 1C shows the log 2 expression ratio values of the genes under different conditions. So here in this table you can see table 1C, you can see log 2 relative measurement C1 divided by C4 C2. So this is the logarithmic transformation in which we have taken the base 2, logarithm of base 2. So here in the rows you can see these are the different genes, and in the column you can see these are the different conditions that is normalized by the C4. Okay, so this is called logarithmic transformation. Okay, so genes which shows greater than 0, the expression of greater than 0 that are upregulated, and genes that shows the expression below the 0 that are downregulated. Okay. Now come to the discrete value, table 1D. In the table we have seen another way of representing information is to convert to discrete numbers the values in the tables mentioned above. Okay, I will show you that. In the case of converting absolute measurement to discrete number, a binary expression matrix of 1 and 0 can be used, where 1 means that the genes is expressed above a user defined threshold. Okay, and 0 means the gene is expressed below this threshold. Okay, above and below. Okay, so in the case of making the relative expression tables or log 2 expression ratio tables discrete, values can be divided into 3 classes, means +1, 0 and -1, where +1 represents gene that is positively regulated, means highly regulated. Gene shows the positive expression, it shows the expression. 0 represents a gene that is not differentially regulated, doesn't show any change in the expression of the gene. and -1 represents a gene that is repressed, means it is down regulated. So these are the three discrete values which we have taken, means genes that shows the expression above 1, we have mapped it to 1, and the genes which show the expression below 1 and greater than -1 that are mapped to 0, and genes that show the expression means that shows the expression below 0 that are mapped to -1, okay, or the -1, okay, that are mapped to -1. So with the table we can easily understand. In the table 1D you can see these are the normalized value, and we have taken the different genes A, B, C in the different rows. So in the columns you can see 0, 1, 0, these are the values. So this is the value like C1 divided by C4, like 10 divided by 20. Okay, here it is transformed. This is a relative measurement, but it is shown as a 0. Okay, because it is less than the 1. Now here you can see this value is 4, like this value is 4, and here it is 2. In the base 2 means logarithmic transformation, it is 2. In the table 1C it is 2, in the table 1B it is 4. So this is the value that is greater than 1. So it is shown as 1, discrete value 1. Now 0, because this C3 divided by C4, so now it is 1, because okay, so it is shown as 0. If the values is greater than 1, then it is shown as 1. So okay, so values that are mapped between +1 and -1, so that is all shown as 0. So this is the discrete value. So here again you can see this value is the -1 in the 1C table, so it is shown as 0. This is 0, this is 0, and this is 1, this is also 0. Okay, so you can see the wherever 1 is coming, 1 means if the values is greater than 1, then it is represented as a 1. So these are all three classes: 0, 1, -1. +1 is for the upregulated gene, -1 is for the downregulated gene, and 0 that means gene that doesn't show any change in the expression. I think now you have clearly understood this. So from here you can see +1 and like here binary expression matrix of 1 or 0 can be used, where 1 means the gene expression above a user defined threshold, means 1, whatever threshold we have taken. 0 means the gene expression expressed below this threshold. In the case of making relative expression table, like you can see here, +1, 0, -1, where +1 represents gene that is positively regulated, 0 represents a gene that is not differentially regulated, and -1 represents a gene that is repressed genes. The process of making the value discrete loses a lot of information, but this is useful to analyze expression profiles using algorithms that cannot handle real value expression matrix, because it is very difficult to analyze the real expression value. There are some method. We feel the difficulty in the analysis of that data. So there it is very useful. It is easy to map between this space like -1, 0 or 1. It is easy. For example, algorithms calculating mutual information between genes or samples. So in that case it is useful if data is too large or data shows the different values. Sometimes it is 4,000, 2,000. So values that are above the 1 we have considered that is as a 1 if n. Okay. So it is easy to analyze. So table 1D shows the discrete values for log 2 expression. So in the table we have seen, if the values is below the 1 to -1, so that are mapped as 0, and if the values are below -1, that are mapped to -1. So now you have seen how it has become easy to analyze. So it is clear now. Now class is over.